Our next presenter, Julien Messonnier from Catalent, who is the Vice President of Service and Technology, is going to present Optiform Solution Suite Bio. So with that, Julien.
So first of all, I'm very happy to be there with you today in order to introduce you a new Catalent scientific offering, Optiform Solution Suite Bio, that pertains to the non-invasive delivery of biological candidates. So this offering really well ties into the visions that define Catalent as a technology company, meaning by working with Catalent, our customer molecule will be screened by all the most advanced technologies to be sure that they benefit from the better market outcomes. So as I mentioned, this offer enables rapid selection of biological candidates for their ability to be delivered on an invasive basis through two biological barriers, either the sublingual or the duodenal barrier, and at the same moment leveraging a set of advanced delivery technologies. So this is also set on strong scientific foundations based upon the learning we gather through the Catalent Applied Drug Delivery Institutes and our non-invasive macromolecule delivery consortium. So we're going to speak about that now. Catalent Applied Drug Delivery Institute is an organization that aims to improve outcome for patients, leveraging and expanding the utilization of advanced delivery technologies. It is an organization that we set back in 2012 under an impulse of our Chief Technological Officer and academic and industry experts that have recognized the gap that exists in collaboration between academic and industry experts, and also in order to foster the development of innovative delivery technologies. So our board membership is made with renowned drug delivery experts in the field, either for the delivery of biological candidates of small molecules, and adjuncted with key opinion leader that we bring in some selected topics. So some of the outcomes and some of the specific action we'll be taking on are the non-invasive macromolecule delivery consortium. This is a consortium for which the value is shared to patients, and value proposition is shared to patients, also to industry experts and to the participants from the consortium. What is it? It is a group of experts gathering on a regular basis that aim to share the knowledge around non-invasive macromolecule delivery. It's made of 4 working groups chaired by industry experts and academic experts, and those 4 working groups pertain to the oral field, to the transdermal field, the ocular, and also the pulmonary field. So the outcome that we've been generating with those are multiple. The first deliverable we had were to generate a set of supportive elements and supportive materials for generating education in the field. We've been issuing a non-invasive macromolecule delivery guide that's providing a comprehensive review of what are the technological approach and the scientific foundations around delivering on non-invasive basis peptide and protein. So this is a very good synthesis of what's the status of the work in the space. And secondly, what we're going to host next February, we're going to host a 4-day conference that's being co-chaired by industry experts, Keenan Park, Randy Mersney, in order to, over those 4 days, under the coverage of the Zinc conference, cover the latest outcome and the latest deliverable from those groups and what have been the new science generated in the field. So you'll be pleased to attend if you wish. So now that we spoke about the scientific foundation, let's speak a bit about the offering in itself. The offering is an offering by which, in a 3-week period, you can assess the potential that a molecule candidate has to be delivered on a non-invasive basis. Leveraging also some unique technologies that leverage some generally recognized as safe ingredients and formulation components. So we can speak about the challenges and the benefits that pertains to delivering those molecules on an oral basis, right? All of you knows that biological candidates with unique safety and toxicity profiles can benefit for longer term indication if they deliver on a non-invasive basis. But also, we can expand on that a little later. But the main focus today is to let you understand what are the key challenges that one face when stepping at the early stage of drug product developments and aiming at bringing a biomolecule to the non-invasive basis. So, number one, there's no single technologies that enable you to resolve those hurdles. Currently, there's a lot of technology out there on the marketplace. There's a lot of one-off companies that have their own approach to delivering those molecules on a non-invasive basis. However, each bears their own set of hurdles and challenges. So in order to maximize chance to succeed and avoid to go in a sequential approach of delivering biological compounds with non-invasive technologies and then go to a trial and error approach, there's a need for a screening platform that enables you to get the right selection and get the right technologies evaluated in parallel. So the second element, those set of technology, they all bring novel ingredients into consideration that are permeation and enhancers. And those novel ingredients, they bring the old set of clinical and regulatory complexities that sometimes lengthen the time taken to bring the product to the marketplace and to the clinic. So why not bringing those with more standard formulation approach and generally recognized as safe ingredients? We're going to cover that later. Also, from the learning that we gathered from the focus groups and from the experience at Catalent, we knew that the biomolecule structure is often the #1 hurdle to absorptions. Once all this technology make the job of opening the barrier, either the sublingual or the duodenal barrier, then the molecules still need to permeate. And we've seen that the molecule structure is responsible for a broader percentage of the chance to succeed. So giving you an example, let's step back in the 90s, sitting on a small molecule business, right? Before combinatorial chemistry is taking a step up. So there was no criteria and no drugability factors that were set there to select a small molecule to be able to deliver orally. And then all of a sudden, trend was raising in poorly soluble drug coming there in the portfolio of companies, and then drugability factor are being factored in. And here, we're convinced at Catalent that there is a need for a screening model to assess those drugability factors that will render those molecules not to be formulated by brute force to the injectable routes, but to be selected on a structured approach with selection factor to be delivered orally. And we're going to show you examples. So, and ultimately, this screening approach needs to be a low amount of material. Milligram level are available at the early discovery stage, and this is also a consideration in the design of the offering. So what we have designed as Catalent is an integrated offering that's designed to rapidly screen the potential that the biomolecule has to be delivered to two barriers, the sublingual and the duodenal barrier, leveraging two unique technologies that are market proven with already oral peptide on the marketplace, and for which we are utilizing generally recognized as safe ingredient. The Optigel biotechnology, which is a lipid-based soft gelatin capsule formulation, and the other one being the Zydis biotechnology, a unique fast-dissolving wafer that releases in a matter of 3 seconds the contents of the tablet into the GI tract, into the sublingual cavity. So, what is the Optiform Solution Suite bio setting? So, it's a set of 3 steps. First step, 3 weeks. In 3 weeks, the aim is to assess with surrogate formulations, very easy formulation surrogates, the ability that the molecule has to cross the sublingual barrier or the duodenal barrier. At the same time, compare these targets, compare this exposure with the target that's set by the partner, and be able to re-engineer very quickly the molecule attributes based upon its ability to cross the barrier. Then, once we've proven whether the sublingual barrier or the duodenal barrier is the right one to target, and that the molecule has some potential, has some legs, after its 3-week screening in animal model, then the next step is to utilize formulations. Quick formulation prototyping in a 4 to 5 week series, 2 to 3 series of 4 to 5 week formulation findings with either a Zydis bio-lyophilase tablet or a Lipid Systems leveraging Optigel biotechnologies. And then you reach quickly the proof of concept in upper animal species on a very fast basis that your compound has the ability to be delivered on a non-invasive basis. So we wanted to facilitate the access for this, and this is the way we designed the program. So first step 1, it's all about setting the baseline. In other words, has your compound some legs to be delivered orally? So number 1, you see many technology exists by which you utilize a variety of permeation and enhancer. Sometimes they are difficult to formulate, sometimes you need to make a tablet, you need a gram, you need 6 months to get a prototype ready. What we wanted in 3 weeks to take a surrogate formulations. We know that sodium caprate is well known in literature to open a high concentration that are not realistic for dose form, the tight junction in order so the biomolecule to come across the tight junctions. So these studies from Tsukibayashi and also so the potential that sodium caprate has to open the tight junction. On the left-hand side, you show the FETC dextran permeation across MDCK cell line that have been selected because they overexpress tight junction protein. And you see with sodium caprate, there's no permeation. And the dark curve with triangles show that after sodium caprate, these cross easily across the tight junction. So, and on the left-hand side, we've been freefying, solubilizing the cell layer and recovering the tight junction protein on the non-soluble fraction. And what we see easily is that further to the utilization of sodium caprate, the dark bar, there is a loss of some of the tight junction protein, claudin and occludin, from the non-soluble fraction of the lipid raft. And this is confirmed by this immunofluorescence assay study, where you see that the tight junction protein are located at the interstice between the cell, and the second set of columns shows that upon action sodium caprate, there is a translocation of the tight junction protein in the inner cell. So we know that sodium caprate is the right surrogate. So the way we screen formulation easily is with an animal model, intraduodenal model in rats, by which we deliver directly in the duodenum by surgery the enabling formulation, surrogate formulations to the animal and measure the exposure that get there. And this is a series. This is a series of various biological candidates we screen of different molecular weight. One must think that as part of the drug ability factor, molecular weight is the main hurdle, right? So we've been ranging for small peptide, very small peptide, up to very large protein domain, some known as antibody mimetics domains. And we've seen that surprisingly enough, there's also some legs for getting some large compounds across the enteral site. And also with the same series, if you compare compound 4, 5, and 6, you'll see that they have similar molecular weight profile, but the way they design, when the tight junction open, they're permeate to different proportions, right? So this is a key learning argument. So when you got a biological candidate at the early stage of development, it's worthwhile to get the right fast and screening technologies to get this happen. We do this activity with less than 100 milligram of API for the duodenal barrier assessment and around 500 milligram for the duodenal barrier assessment. It's a fixed price offering also. So the second stage is enhancing. So we're going to focus at that stage on the uniqueness of the technology that we bring there. Number one, the optical biotechnology targeted for duodenal delivery is focused on, number one, being enteric-coated multi-particulate units that are including GRAS ingredients, lipid ingredients. And through the works we made since many years on bringing over 50 polysoluble entities to the marketplace, we've been noticing that some of those lipid systems have the ability to fluidify the membrane, the cell membrane. And also at the same time as it resolves poor solubility hurdles, it also do resolve for certain small molecule permeability hurdle. So that's when we started in 2008, a research work funded by the French government that have led us to the identification of the unique formulation component setting that enable, with generally recognized as safe ingredients, to fluidify the membrane and get it in high level of concentration at the point of delivery. So there's also some other advantage to the optical biotechnology that you're going to find on this slide. We're not going to elaborate today for the benefit of time. The second technology we leverage for sublingual delivery. If there is a hit in the assay phase for getting the right target exposure with the biomolecule by sublingual delivery, we work with lyophilized tablets. Zydis biotechnology is the technology that's applied for the delivery of desmopressin, the potent vasopressin analog that you know right now on an oral basis, despite a fairly low exposure. But this is indeed, in 3 seconds, releasing the field content, the capsule, the fast-dissolving tablet contents below the tongue, and generating high concentrations of the molecule with or without permeation enhancers. And we've seen positive outcome from those studies. The other advantage of this processing technology is a processing technology that's well suited for peptide and protein and others, because it's a lyophilization technology. How does it work? You solubilize the peptide and protein in a matrix, in aqueous matrix. You fill this into a blister cavity, and you immediately lyophilize it to get the unique disintegration property of the lyophilized wafer. So that's another embodiment, another advantage of this technology going forward. Now we're going to bring you a couple of examples. So salmon calcitonin is the first one. And we've been comparing that salmon calcitonin data to the Oracle 200 microgram formulation developed by Tarsa using the existing technologies on the marketplace, which is a monolithic tablet. So what this slide shows is in dog studies. How we go. So we got the baseline sodium caprate formulation. Maybe I can show it on screen there. This is the, all right. This is the sodium caprate baseline. So salmon calcitonin has shown that with submission quickly to sodium caprate, it has the legs to be delivered on a non-invasive basis. This exposure ties into 1 or 2% exposure, whereas the Tarsa formulation goes around 4 or 5. And then we compare it with a lyophilizase that had been directly given by oral gavage in the GI tract, bypassing the sublingual quality. So there's no absorption. There's no exposure there. However, when the lyophilizate is delivered to the sublingual cavity and integrate in 3 seconds, that's where you got the target exposure that you got with sodium caprate formulations. And in parallel, we'll be screening those with different type of lipid formulation. This is the green curve, the blue curve, and this is the purple one. And we see that the exposure we got overpassed the current exposure we got with the TARSA technology. So that's a good convincing argument of the parallel screening approach of different technologies and how it could be suited for certain drug candidates upon specificity. So the next part of the presentation is how do you move from a concept to a final dose form? Because at the end of those studies, you're going to get a lipid systems that's purely a lipid solutions. And you're going to get also at the other end a lyophilizate. How do you make a final dosage form? So this is a macromolecule that we've been screening that's extremely poorly permeable because it's extremely strongly negatively charged, and occludin and clodin being also responsible for the charge selectivity of the tight junction being negatively charged. This was a negative embodiment for the molecule to be tested. This molecule also was a good candidate because the subcut injection was providing complete absorption, but also it has a pretty wide therapeutic index. So what we've been doing, we've been bringing the compound with caprate to dogs, and we see that the baseline is 20%. And when you provide the lipid formulation, the optical bioformulation in the GI tract, in the gastric juice, you reach 10% exposure. However, when you bring it into an enteric-coated softgel monolithic unit, you reach 40% exposure with this compound. However, the individual curves are pretty large. And some of you which are pretty knowledgeable in the field of non-invasive delivery will know that it's a common setting and common embodiment for all those technology. But we wanted to investigate why there is the high variability. So we've been tagged the capsule content with iodine derivative, contrast agents, omnipaque, and we've been placing those, delivering those to dogs, and at the same moment doing CT scan study to locate where the capsules stand. So the first series of experiments shows that the monolithic capsule stay in the pyloric antrum and they never reach the duodenum because of steric hindrance, and this leads to the low exposure system. And by analogy, what you find with the other alternative technology that you get from the other competing technology, we got a Casma or Unigen and others that are large monolithic units, that's a common setting that you find. But surprisingly enough, other specific individuals see the capsules that progress immediately in the duodenum, and that's where you reach a target exposure beyond the 30% that we've seen. So by analogy, what you're going to get with enteric-coated systems, a monolithic system is a 0 or 1. Because of the size exclusion criteria, you're never going to get, either you got exposure or you don't get exposure. If it passes duodenum, you get exposure. If it don't pass duodenum, you don't get exposure. So that's the reason why when you develop nowadays a proton pump inhibitor, nobody's going to develop it with a monolithic system. You're going to develop it with multiparticulate units. So we've developed for each of those technologies some multiparticulate unit systems that are ensuring that the population of those small mini capsules, enteric-coated or colonic-coated, pass the GI tract, reach the stomach, and deliver the right exposure to the site of absorption. So that's a quick review of what the Optiform Solutions with Bio offering is. It's said about our previous experience with various peptides we have in development. We've got 13 peptides in development right now at different stages of preclinical and clinical development. And we have also 2 commercial products that are peptides that are already delivered there by Zydis. So this is the desmopressin formulation, and the other one is a softgel product. So in other words, it's an easy process that integrates 2 enabling techniques with generally recognized as safe ingredients, and you can get, for a limited level of API, few hundred milligrams, for a small period, 3 weeks, the proof of concept that your molecule has some leg to be delivered on a non-invasive basis. And then afterwards, you can get some trust and judgments to propel this or not to the phase 1 and first in human study. So thank you for your attention, and we'll welcome any questions.